Hello, my name is Iya Wang. Today I will be presenting my research project testing fungal prohibitory properties in some common and uncommon food items. But first and foremost, I want to thank my mentor and professor, Dr. Christine Case, for inspiration and ongoing support on this project. Before I outline my experiment and report my result, I would like to highlight the significance and the motivation for this study. Over 700 million people are estimated to not have enough food and lack proper nutrition across the world, which is to say 700 million people go to bed hungry each night. Well, our society has been growing technologically and economically at an unprecedented rate. The amount of starving people have been rising since 2014. At the center of this issue is food waste and food spoilage. Approximately one-third of the world's food supply is wasted, with food spoilage as the major contributor. Seeing fungal growth or mold is the number one reason people throw away their food. There is an urgent need to protect our food from spoilage so we can decrease the number of people that go to bed with an empty stomach tonight. Fungal spoilage is also costly. Studies by the United States of Agriculture show that $162 billion are lost each year to food spoilage in the US. So we see the urgency and vitality for us to find novel ways in which we can address the issue of food spoilage in cost-effective manner with low potential complications. Fungal infection that leads to food spoilage is carried out when fungal spores spread to new uninfected foods. Fungi are chemoheterotrophs who derive all of their metabolic energy from the breakdown and consumption of other organic matter, for example, our food. Once the spores have found a sufficient host, they will grow into hyphae and mycelium that produces the common look of molds we're used to seeing. These hyphae can then form sporangium and produce additional spores to infect other foods. Common fungi that infect our foods include Alternaria, Aspergillus, Penicillium, and Rhizopus. The current methods to prevent the growth of fungi and food spoilage are listed on this slide. But there are some limitations to these methods, including higher cost, altered tastes and textures, reduced access to resources, and potentially harmful chemicals. Due to these limitations, we must find alternative low-cost ways to help preserve food, especially in countries that do not have access to the resources for maintaining preservation. I propose a strategy where we can use natural treatment solutions derived from herbs or other food items that are already safe to consume. Previous research has shown the antifungal properties of some herbal extracts, but many of these studies focus on the antifungal properties against fungi that can infect humans, but not food. So here I test the antifungal effects of 14 different food on Fragaria, commonly known as strawberries, a fruit that has a low shelf life and spoil easily due to fungal infection, but which makes them the ideal substrate to use for this experiment. Next, I will outline my experimental design. In this table, you can see the list of treatments I used. The negative control was the strawberries sprayed with tap water, and the positive control was clotrimazole, which is an inedible, over-the-counter antifungal cream, commonly for treating athlete's foot. Please note that the two treatments with an asterisk were bought pre-extracted with distilled water and used as is. Most of these treatments are readily available at Asian supermarkets. These are common food items in China, and I have eaten all of them, except for the greenwell roots. The guayusa and moringa are tea leaves I brought home from Ecuador. To prepare for my treatment, I first grind it into powder using a household spice grinder. Then I mix the powder and water heated to 50 degrees Celsius at 1 gram to 15 milliliter ratio. I used 5 grams of powder and 75 milliliter of water for all of my treatments. Next, I let the mixture sit and brew for 12 to 48 hours before I strain it through a coffee filter, so that the final treatment solution can be added to a clean spray bottle to be used on the strawberries. Then I prepare the strawberries for inoculation and incubation. I used a seedling starting pan with a heat mat and humidity dome as the incubator. The heat mat was covered with a regular bath mat to diffuse heat. Then I cut the strawberries in half and placed them on a coffee filter with the cut surface facing up. Then I sprayed 10 times, which is about 1 milliliter, with each respective solution. Because each pan fitted 8 coffee filters, I was able to test for 2 different solutions 3 times in one dome, and using the 2 filters left for positive and negative control. After 30 minutes incubation at room temperature, each strawberry was inoculated with fungal spores using a mixture of tap water, mold spores, and soil in my backyard. I dip a Q-tip in the dirt water suspension and wipe across the cut surface of the strawberry halves. Then I cover the seedling starting pan with inoculated and treated strawberries sitting centered on the coffee filter at temperatures of 75 to 85 degrees Fahrenheit, which is about 23 to 29 degrees Celsius. After 2 to 3 days, the strawberries grow mold, and I took pictures of each strawberry to evaluate the percentage of mold coverage by overlaying a transparent graph paper grid, as shown here. Then I evaluated the mold samples under a microscope. I tested each treatment at least 3 times and averaged the results to look for statistical significance. My results show that 4 treatments caused the strawberries to mold more than the negative control strawberry did, while 10 treatments caused strawberries to mold less than this negative control. Out of the 14 treatments, only the clotrimazole cream and the groom-well root showed a statistical significance. The groom-well root showed a p-value of 0.04 and a mean percentage mold coverage of 14%. There also was a high variability between each replicate, as shown by the large range of arrow bars, indicating possible confounding variables. This can also be seen in the negative control. After assessing the mold spore arrangements for each moldy strawberry under the microscope, I found that Aspergillus and Rhizopus are among the most common genera of fungi that infected the treated strawberries. And the treatment that did not show a certain genus of fungus could potentially signify a species-specific fungal inhibitory action. To conclude, all the treatments did not work to inhibit the strawberries from molding. While the groom-well roots did show a potential for fungal inhibition use in food preservation, further studies are needed to confirm these results because of the limitations of home experiment, which made controlling for an even growing condition very difficult. So I will further investigate once we're back on campus and able to use a growth chamber to better control the confounding variables. Using tap water could have also affected the efficacy of my negative control, as there is a small amount of chlorine in our drinking water. Future studies should use distilled water as control and quantify inoculation with known fungal species. Fungi have been with us since the beginning of time. Their presence is crucial to our survival, but because of modern society's higher standard of living, food safety-related regulation also lowered our bar for judging when to throw our food. Food mycology is a young but important science, as what we discover here could make an impact on the society, from preventing food waste and the associated economic loss to saving millions of people from hunger and improving human health. And that concludes my presentation. Thank you very much for your attention. I'd be happy to further discuss with anyone the details of what I presented here. Feel free to email me at iyawang@my.smccd.edu.